Lesson No 14 DNA Translation. Protein Synthesis. DNA Transcription. DNA can unzip itself and RNA nucleotides match up to the DNA strand. Both DNA and RNA are formed from nucleotides and are called nucleic acids. DNA replication, protein synthesis, transcription, translation, cell cycle replication, DNA and transcription. Translation. Protein synthesis, how DNA is copied, replication. How information in DNA can be used to make a protein. Every 3 mRNA nucleotides, codon, specify an amino acid. Protein synthesis. Prokaryotes. Transcription and translation occur in cytoplasm. Eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. Genetic translation, the synthesis of polypeptides on ribosomes. Translation is a process by which the genetic code contained within a messenger RNA, mRNA, molecule is decoded to produce a specific sequence of amino acids in a polypeptide chain. It occurs in the cytoplasm following transcription and, like transcription, has 3 stages: initiation, elongation and termination. In this presentation we will look at the components and stages of DNA translation. DNA translation. The cell uses information from messenger RNA to produce proteins. Components of translation. The key components required for translation are mRNA, ribosomes, and transfer RNA, TRNA. During translation, mRNA nucleotide bases are read as codons of 3 bases. Each codon codes for a particular amino acid. Every TRNA molecule possesses an anticodon that is complementary to the mRNA codon, and at the opposite end lies the attached amino acid. TRNA molecules are therefore responsible for bringing amino acids to the ribosome in the correct order, ready for polypeptide assembly. It is important to know that a single amino acid may be coded for by more than one codon. There are also specific codons that signal the start and the end of translation. Aminoacyl TRNA synthetases are enzymes that link amino acids to their corresponding TRNA molecules. The resulting complex is charged and is referred to as an aminoacyl TRNA. Initiation. For translation to begin, the start codon 5OG must be recognized. This is a codon specific to the amino acid methionine, which is nearly always the first amino acid in a polypeptide chain. Elongation. The polypeptide chain is built up in the direction from the N-terminal, methionine, to the C-terminal, the final amino acid. Termination. One of the 3 stop codons enters the A site. No TRNA molecules bind to these codons, so the peptide and TRNA in the P site become hydrolyzed, releasing the polypeptide into the cytoplasm. The small and large subunits of the ribosome dissociate, ready for the next round of translation. Transcription/translation quiz. Why is transcription necessary? Describe transcription. Why is translation necessary? Describe translation. What are the main differences between DNA and RNA? Using the chart on page 303, identify the amino acids coded for by these codons, UGC. Quiz. Translation of DNA. Where in the cell translation occur? Cytoplasm, nucleus, mitochondria, Golgi apparatus. Which codon enables initiation of translation? Org, arg, tat, gug. Thank you for your attention.